So thank you so much for joining us today, Gabby, and for offering to give us this talk. Dr Gabriella Lindergard is a clinical lecturer in nursing at the University of Manchester and Manchester University NHS Foundation Trust with a PhD in infection and immunity. Drawing on her journey from research nurse to chief investigator, she brings a unique blend of academic expertise and hands-on clinical research experience in infectious diseases. Her work focuses on innovative, patient-centered approaches to HIV and gut microbiome research, bridging frontline practice with cutting-edge science. Okay, I'm going to stop sharing and hand over to you now, Gabby. Thank you.
Thanks, Jude. That sounds amazing. So I'm going to share my screen. There. Is this okay? Or should I put it in?
Yeah, it's showing at the moment as slides, but not like present, not
like presenter.
Yeah, you know, not slideshow view.
I'll do. It always covers that for me. Yeah, move it. Covers the icon. There it is. Slideshow. Oh, just slideshow. There.
Perfect.
Is that okay? Yeah.
Yep. Looks good.
Thanks, Jude, and thanks for making my intro slide a lot easier. And thank you everyone who's kind of chosen to come and listen for a while. So I'm on a integrated clinical academic fellowship with ICAT, which means that I am 50% with University of Manchester, so I'm actually employed by University of Manchester, and with a honorary contract with MFT. A few years ago, one of the matrons at RNI, she was on the 70 leadership, NIHR leadership program, and she managed to start her work with collaborating with the university so that there would be a bit of funding left. So the ICAT is normally, well, it has been for the last, well, I think it's been on for about 10, 15 years, mainly for medics and dentists, but the matron who was on the 70 program, she managed to negotiate a bit of the leftover money for a 3 posts, one in infectious diseases, one in pediatric, and one in cancer research. And I applied for the infectious diseases one. So as I said, I'm 50% at the hospital, at the Infectious Diseases Research Department at North Manchester General Hospital. I got a bit of time for research, which mainly I've spent on delivering studies within the team. I'm a PI for a few ID infectious diseases studies that we deliver there. I'm also the North Manchester General Hospital local lab manager, and I'm the lead research champion for RNI. And then my other 50%, which also incorporates quite a lot of research time, which gave me the time and, well, basically give me some peace to sit and work out a protocol. And so I would be the study coordinator and writing the protocol for research proposal. Then I also have a few master's students, and of course I need to do quite a bit of presenting and spreading what we're doing. So the aim with these the ICAT is to give you some time and training, so coming away from the clinical work a little bit, but we also obviously hope that it's going to lead to some external clinical academic funding. Just in that change. Change my stock. To change, go for the next slide. Nothing happens. This can I do? Escape doesn't work either. No. This is exactly what you don't want to happen. Okay, gonna see. Does it? I don't know. So this is my long and winding road. I started off with doing biology at the university, and then actually simultaneously I did a nursing degree. And I think I knew quite early on that it was infectious diseases. That was my my main interest. So after my registration, I worked in infectious diseases ward. This is back in Sweden, where I'm from. It was also a tropical medicine ward, and the part of that, don't ask me why, but the parasites side of the tropical medicine was, I thought it was absolutely amazing. I think it's because the parasites and humans are kind of evolved together for such a long time. So there is a relationship that maybe you don't think about that often, but humans have evolved parallel with parasites and almost incorporated that into our healthy life. And I thought that was very fascinating. So when I found that you could actually do a whole degree in parasitology in Glasgow University, I took the chance straight away. There was often when I talked about I was doing parasitology, I often got confused with parapsychology. So it's a bit funny that people knew more about parapsychology than you do in parasitology. Anyway, I continued there, and then I went to Liverpool, the School of Tropical Medicine, for a research master's where I did quite a lot of field work. And then back to Glasgow for a PhD in infection immunity. Then I, as you do, continue with a few postdocs with a little bit of a break to, I had 2 boys and decided to stay home a little bit with them. And then back to postdocing again. And then when the family made a move to Manchester, I saw opportunity to kind of change the direction a little bit and applied for the infectious diseases research nurse post, where I then became the lead research nurse for about 10 years, so the last 10 years. Well, and then onto the integrated clinical academic program as I am now. Let's see, this is good. Oh, I did hope. So yeah, I did feel a little bit like I was living parallel lives. I was having my science part in one way, and then I was having my nursing, and I did try, and it's a, well, I can say now, it's a shame that it was so hard to combine it. I did have made, especially volunteering to do quite a lot of field work. I felt during my master's project, that's when I felt like I was kind of using my clinical background a little bit more. But then as you go into more lab-based, you go further away from the patients. So I feel now that I'm finally, after quite a long time, a long and winding road, is a bit more using both of my science and my clinical nursing degree. As I said, I took every opportunity to get more involved in the field work. So we did quite a lot of malaria studies, looking for new diagnostic methods. We did some deworming programs. And as I said, it was quite useful to be able then to both do the science, and I could take the blood, so I can do the examinations, and so we didn't need having a separate person to do that. I wasn't sure if we were going to have this slide in there, but of course, hurdles always happens. For me, I think the time during the pregnancy and trying to have some kind of maternity leave was the time where you saw the difficulties for especially women. How it's quite hard to come back for that. So even so, I had my first son in America, and what I got was 6 weeks short-term disability, like I was disabled after having a child. So that was kind of a bit of an eye-opener from the hurdles you can face, and you have to do the best you can, I suppose. Maybe a little bit of a positive thing was that my partner, the dad, he had to take responsibility for a 6-week baby, and can say now that their relationship might be better for it, because he just had to. Moving on from that time, where the advantage for working in academia was, of course, that it was a little bit more the flexibility you have, so that we could share it. There was long days, just handing over the baby to the other, but it did work out, and I think it worked out for the better. Coming back, expecting my second one, was realized that my whole postdoc salary was going to go to childcare, and that's when I decided to be home with the boys for 4 years. It was all together, and then trying to come back after that was a challenge. There were supposedly grants that you can apply for, like career re-entry fellowships, but they were not really specific for women who have been home for a while. You ended up actually competing with everybody else anyway. So as I said, I did work in all sorts of areas. My PhD was in infection and immunity, working on trypanosomes. So the single-cell organism that's causing sleeping sickness is transferred by tsetse flies. And then it might seem like it's a bit all over the place, but the methods, like the molecular genetic methods that you're using, can be fairly similar, but depending, even though you might work on different organisms. So after my PhD, I did a postdoc in working with cryptosporidium and trying to find new novel diagnostic methods, which was that was in upstate New York. Very haven't really thought about it before, but the whole water supply to New York City is upstate New York. I'm surrounded by cows, which then, of course, often have this cryptosporidium cysts in their feces. So if that would come into the water, the whole of New York City would be infected. So we were trying to actually work out for different kinds of species. They look exactly the same, but they might not be as infectious to humans as you might think. Then I, apart from trypanosomes and cryptosporidium, the last few 2 postdocs was all about malaria and trying to find new targets for vaccine targets in some of the surface proteins on the red blood cells that the malaria parasite expresses. So as I said, infectious diseases research department, I was for 10 years. We were was a very, very active department. During those years, we delivered over 100 clinical studies. We helped to set them up. We delivered them, and we were maybe the best part of it, or the most interesting part. We were delivering a lot of hepatitis C treatment studies. So before, and as I was working for the infectious diseases wards in Sweden, there was no treatment at all for hepatitis C. But during this time, there was a new development of medication. So for a while you tried a horrendous 1 year, often up to 1 year treatment with interferon, where the poor patient basically felt like they have flu for a year. So it was quite a few who didn't even finish off their treatments because they just felt so bad and wasn't worth it. But after there was a few companies that tried different treatments, and now we can treat hepatitis C within 6 weeks, and actually treatment, so it clears the virus altogether with some of the new drugs that we tried as fosbuvir. But not only, so we did, there was all sorts. There was HIV, there was hepatitis, TB, C. diff, any kind of infectious disease that you can think of. So when you think about it, when you work as a research nurse, you get a lot of experience in setting up studies. You can see how the protocols and the ethics, how it works, what it looks like, what Irish form looks like. So you're quite in a very good position to identify research needs and kind of know how to get things started. Because you've seen so many different ways that it can be done, and it's almost like a missed opportunity for research nurses not to get involved in doing their own research, because you see what's needed, you meet a lot of patients, you see what the gaps are, and as I say here, the nurses are often in the ideal place to identify research needs. So NIHR is a government funded, which means that it's publicly funded. So therefore you can see how important it is to involve the patient and the service users really from an early start. And not only is it because they help to pay for your research, there are actually lots of really good like you get the help, from the patients to write your protocol, because you feel like it's more relevant when you've had the feedback from patients. And as I said before, how you see you're likely to reflect the patient's needs much better, and if you can have that relationship with patients or service user from the start, it kind of help you to shape your research question, and also they can be like a soundboard for you when you're writing your protocols and the patient information sheets, and you can kind of show and get feedback, and that would help you as a researcher as well, because it will help with it if it's well understood, but the ones that's going to hopefully benefit from your research, your recruitment is going to be easier, retention is going to be better, and you building that transparency already from the start. So there's, I'll put a vocal here because that's one of Manchester MFT's big collaboration with the vocal team, where we help you with patient involvements, and if you need how to get started and how to get the contact, and they're very, very helpful. So we coming on a little bit to my research now. My research area very much developed from questions that the patients had. They were asking questions, but we didn't know the answers. And then you were thinking, why do we not know these answers? What's missing? What could it be? How can we kind of form a question? So as I said, we worked quite a lot with HIV new drugs, and so when they came to the infectious diseases research department, we looked after all of their treatment, so they didn't have to go to others, because they stayed often they stayed in the trial for several years. So you were meeting them often, once a month or even that. And quite often when they changed their antiretroviral treatment, they were asking, so they would say, oh, they're starting to get GI problems, they were putting on weight and things, and they were, and they would ask, why was this happening? Why is there something to do with my drugs or not? And that information wasn't available. So coming on to my special interest with the gut microbiome. So a little bit, so when you're born, you don't really have any bacteria, but as soon as you're born, the gut started colonizing. So how is it determined what bacterial colonies you're going to have in your gut? The first determinant is actually the way you're born. So cesarean versus vaginal birth, their gut microbiome in the beginning is actually really different. You can actually divide them into two different paths after that. And then the next step, with infant feeding, are you going to breastfeed, are you bottle feeding? That also affects the first colonization of your gut microbiome. Whatever the life cycle stages, so with your age, there's a lot of change in the beginning and in childhood, and then it got a bit more stable. And then you got a big change during menopause for women, and then in older age. Your geography, wherever you live, the environment has a huge difference. That's kind of what you. It's very hard to say what a healthy microbiome is, because healthy can mean so many different things, and it is probably not like one specific. Like one specific set of bacteria that you can say these are the good bacteria, these are the bad bacteria, because it kind of many different bacteria can do similar things. So and so sometimes it'll be a bit hard comparing the gut microbiome between different geographic places. So what's healthy in one place, their microbiome can be completely different from what's healthy in another place. The food offers to have a very huge effect on you. The other kind of stresses, exercise, metabolic and psychological, can actually change quite a lot of bacteria what you have. And then just as much or even more than the food, all sorts of drugs have a huge effect on your gut microbiome. So obviously antibiotics is the one that changes the most, but even just some of your normal pretzel or other things for your stomach actually wipes out quite a lot of your gut microbiome as well. So it's just to show that it's the complexity of trying to study what your microbiome does and what it is, because it changes. And not only is it changing between people, but it also changes along the gastrointestinal tract. So you have very different bacteria up in your stomach that you have in your small intestines as you have in your large intestines. And one of the reasons why it is so hard to study is because in your intestines you have anaerobic bacteria. So how do you get them out without exposing them to air and changing it, or they might just die off if you do it? And I suppose the ideal way would be doing biopsies, but that's obviously a little bit harder, or maybe not as ethically right as compared to just collecting stool samples. So why do you have a gut microbiome? There's lots of reasons why you need them, and they're totally they're absolutely needed for a healthy life. They produce, first of all, they've been known for a very long time. They produce a lot of the vitamins that we need. Most of the B vitamins and the K vitamins, and some of the studies just show there's even up to 30% of your daily recommended intake is actually produced by your bacteria, not what you're ingesting. It's obviously helped very well with the digestion, so they are responsible for a lot of the fermentation of food, so that you can easily digest it. And there you can see if you don't have the right bacteria, some of the digestion will actually be harder. So with, say, for instance, with if you're talking about different kinds of fibers, or if you're glucose, okay, if you the overgrowth of the good bacteria then also protects against the bad. It's not very good to use the terms good or bad bacteria, because they have different functions in different individuals. But if you don't have a good set of bacteria, and then you get an overgrowth of the bad ones, where obviously Clostridium is one of the major examples of that, where you knock out all the good bacteria with antibiotics, so that the Clostridium difficile got a chance to multiply and producing a lot of toxins that can be lethal. So these bacteria in the gut, they also produce a lot of different proteins, of the molecules that kind of regulate the immune response. And one of the slightly newer areas of research is the gut-brain axis, where you can actually prove that a lot of the bacteria are producing neurotransmitters and neuroregulators that directly affect your brain. So you can ask, why is there so much talk about the microbiome now? Well, I suppose one of the main reasons is the Human Microbiome Project, because that was possible now with the new technology, where sequencing became much easier, less costly, and so before we could directly sequence the bacteria that we can collect in stool samples, you had to isolate the bacteria, and you had to be able to grow them. As I said before, some of them were, most of them were anaerobic bacteria, so very hard to grow. But with the Human Microbiome Project, there was a few reference individuals who started sequencing, but similar to the Human Genome Project, but this has now moved on to a second phase, where we're looking at the health connections with the specific sets of bacteria. There's a lot of evidence for what modulation of cardiovascular diseases, so that the metabolites that the bacteria produces have a direct effect on your cardiovascular health. One of the first studies was the obese and lean. It was actually fecal transfers, where you could transfer a obese microbiome to a mouse, and causing the mouse to become obese. So I think that was one of the first studies where people kind of, oh, this actually does has some clinical impact, of course, it wasn't as straightforward as you thought it was. But now the first studies were really showing in humans the clinical correlation with microbiome coming out, and this is really interesting. And then the mind-altering microorganisms has given rise to a whole new area of research, psychobiotic. We actually treat psychological and mental disorders with a change of diet. So what we were thinking, when you have an acute HIV infection, it results in a massive depletion of the CD4 T cells. And that destroys the gut epithelial barrier. So what's really important in your gut is that mucus layer that protects you from invading whatever you have in your gut, bacteria or viruses or anything. Yeah. So when a person becomes infected with HIV, there's a massive change in your gut microbiome. When you then start to treat with antiretroviral treatment, that dysbiosis, so the changes, the negative changes in your gut, it kind of it recovers a little bit, but it never recovers fully. So there's always if you look at like big population studies, there you will see there is always a big difference between a population or a large group of people who do not have HIV and the ones that have the virus. But not only that, but the different types of or different classes of antiretrovirals. There's quite a few now. So the integrase inhibitors, the and the protease inhibitors, they seem to have very different effects. But these studies are only done on like big populations, so it's very hard to tease out if the real cause and effect. But that was one of the reasons why we started looking at the microbiome changes in HIV patients. So our hypothesis is that the gut biome, so there's a virome as well as the bacterial, so this is the virus population, is changed by the antiretroviral treatment and causes then alterations in the bacterial microbiome, leading to the clinical effects for HIV patients. So the antiretroviral treatments are developed to be very specific for the virus, but nothing is that specific. So there's probably causing changes in other viruses as well. And obviously, there's always this commensal relationship between bacteria, virus, like our environment. So if you change one thing, you could think that you're changing other things downstreams as well. So this is our study, my study. A prospective observational study of the impact of the gut microbiome in HIV-infected individuals in Manchester, UK, when switching from one class of antiretroviral drugs or delivery method to another class of delivery method. I'm saying delivery method as well because we now have intramuscular HIV treatments. So in that case, you would stop taking oral drugs altogether, and you will just have intramuscular injections. Is every 8 weeks at the moment, and then you can see that. Well, you can think at least that you're totally bypassing the gut in that case, even though you're not absolutely sure that you are. The concentration of the antiretroviral treatment in your gut could still, well, we don't really know. So this was the idea. We will collect serum and stool samples from the patients before and there was a change in their antiretroviral treatment. So there could be lots of different reasons why they would change, but we were trying with very specific inclusion-exclusion criteria so that the only change they had was the change in the antiretroviral treatment. So everything would be the same. We had a questionnaire before and after, so asking about diet and exercise and things that could affect it, and obviously if they were changing any drugs or if they were changing the way, well, we definitely didn't want them to go on holiday in the middle of it or anything that could change your environment and things as well. So we would collect a sample before, as close as possible before, and then after 3 to 8 weeks after the change. And then we would characterize the microbiome, and we would also collect serum samples at the same time so we could look at the immune response and the immune factors. So it's quite a simple, straightforward. We would, it was the identification that was probably the most, the hardest, the most time consuming to try and find people that were, because we didn't want them to, I mean, one of the reasons for changing your antiretroviral treatment is obviously that it doesn't work, so that you will have virus, and we didn't want them to have like a high level of viruses, because that was obviously going to affect the microbiome as well. So it was a time-consuming procedure to identify only the people that had other reasons for changing, maybe simplifying the regimen or something wasn't available, whatever. So we just on day one, we just collected the sample. I did the bloods, we did a questionnaire, and then the same day or the day after, they produced. Wasn't as easy as doing a urine test, was it? So you had to kind of arrange to collect the stool samples the next day. So I come up for 5 minutes. Oh my goodness. Okay. And then between 3 and 8 weeks later, we would do exactly the same. There's our student samples. So now we're getting all the data. The data is dropping in. There's quite a lot of it. And I was hoping that today I was going to show some proper results. So we started looking at well, you start looking at the phylum, you go down to the classes, the orders, your families. So now we're down to the genus level, and we're starting to see some of the differences between the control groups and the groups that had the changes. But we have the species. You get an awful lot of information out of this. I think there's about 10,000 species identified with this the shower shotgun sequencing. So I'm working together with one of the bioinformaticians here at the Lydia Becker Institute to help me kind of tease out all the differences, because it becomes there's so much data. So you need a good bioinformatician to help you to sieve out the important from the non-important. So this is what we're hoping we're starting doing. We're looking at the controls, we're looking at before and after. And as I said before, the microbiome is so complicated, so there's so much variation. So what we really want to see is just, is there a change or is there isn't? And this kind of study hasn't actually been, I haven't seen anything published, haven't been done before, where you compare the person with themselves. So you can do big, large population studies where you have thousands or hundreds of thousands of people, and you can see some kind of correlation. But now we're comparing the person with themselves, and hoping that the only change that happened during that time the change of the antiretroviral treatment. This is where we're really looking at to do now. This is a paper from, you might have heard about Tim Spector and his Zoe apps, but he's one of the first ones to start to look at the markers of nutritional cardiometabolic health with some of the bacterial species. So picking out which bacteria could be linked with health, and which ones can or positively or negatively correlated to health. So we're going to do that as well, maybe even tomorrow, to just as a little bit of a side project. Yes, I don't even have the results started, having written about half of the paper by now, but without the results section. But you had to start thinking about the future and the next because of funding. So I really want to continue to build on the data that we have now, but maybe go into a little bit more about what people can actually do to change in a positive way. So people living with HIV, they already live with a chronic inflammation because you don't get rid of the virus. The virus is always there, so their immune markers are always a bit raised. And if lifestyle behavior factors can really contribute to this level of chronic inflammation, so for people living with HIV is almost like a double whammy. You have the underlying one, and then you have your lifestyle behavior factors on top of that. There's a British Society Lifestyle Medicine Intervention Program that is very successful in changing in a positive way, using different pillars of behavior. And for my next continuing research, I want to see if that can actually help with people living with HIV as well. So the idea is to measure the level of inflammation, both subjectively with questionnaires and how you're feeling, how your mood and sleep and energy levels, but also having an objective measure at the same time. So you're looking at immune factors, you can look at your changes of HbA1c, you can look at the changes of CRP, and then again, if there are going to be any changes in microbiome flora. So this is where I'm at the moment. I try to move on to the next step for career development, looking for funding. I applied for NIHR Senior Clinical and Practitioner Research Award a year ago, didn't get that. I just I spent about 3 months in the springtime applying for NIHR the postdoc award, just heard a couple of weeks ago that I was rejected. So now, got my fingers crossed for the NIHR, the pre-application support fund. And as I said, these the Integrated Clinical Academic Fellowship I'm on now is a 4-year project, 4-year funding up till next June. So it's really time to start finding something else. And because I'm passionate about prevention, health, for what you can do for your health, I can miss the opportunity to say a little bit about gut health, how to do things, have a behavior that contributes to a gut health, and as you obviously heard, how much it can influence your body and mind. So the main thing is probably about the feeding your bacteria with, but the best thing is with the plant fibers. Because the fermentation and of the plant fibers and the byproducts is one of the main things, the short chain fatty acids, that really helps with the mucous membrane and preventing things like leaky guts and things. Probiotics, live bacteria in fermented foods as a regular staple, but there's actually now some evidence that even pasteurized fermented food could have a positive effect. Exercise actually changes your microbiome diversity. So you could measure that in a trial similar to what we're doing here. If you measure before and after, and then you of course have things that most of the processed food could have an effect, and all the artificial sweeteners now seem to change your microbiome. High sugar foods, and then stress and lack of sleep also promotes the kind of wrong kind of microbiome. And I think that was it. Happy to take any questions.
Great, thanks so much, Gabby. If anyone has any questions, please put them in the chat. I have one or two for you. I was really curious, Gabby, you said it was really hard to get back to work after you sort of returned from your maternity leave. And I was wondering, you didn't say how you managed it in the end.
How I managed it? Well, I did, so I was home for 4 years. And then when my little one started in school, I went back to part-time. So I managed to get a part-time postdoc. And then I didn't get any of the grants I applied for, but I managed to basically employed in other research groups as a postdoc for because of the experience I had from before. So yeah, it's not. It wasn't. It would have been great to say that yes, I managed to write the grant and I was successful in the end, but no, it was a slow recovery.
And as well, I was wondering, how have you found it sort of managing your research career alongside, maintaining, I guess, your registration as a nurse? Because especially, I suppose, given that your research is quite different, perhaps, to what you might otherwise be doing in your nursing role.
Yes, I do find that now, because I only have the fellowship to the next year, so there's no like ready jobs for me to apply for. I mean, whoever I speak to say, "Oh, you got these special skills," so of course everybody's going to want you, but there's no ready jobs to apply for. So that's where I feel like I always have to create or change, or it's a shame, because the research kind of thinking and the methodology, it's advantageous for whatever research you're trying to get into, but yeah, and I suppose one of the reasons why I do want to go into more lifestyle changes is a little bit more easier to combine with a clinical career, and you would think that was something that would be, well, I think it would be very useful for NHS to invest a little bit more of prevention, and I think that's yeah, I would like to continue with with more molecular genetics, because that's what I kind of feel comfortable with, but I think I am changing a little bit because of what the need, and you have to change what the need is, of course. Yeah, you need to be a little bit flexible. So I'm trying to be a bit more flexible.
Brilliant.
Thanks.
Okay, one last question. So if there's anyone who's on the call and who is considering starting a research career, but is not sure what their first steps should be, what would you suggest to them?
Oh my goodness. Well, you have to enjoy it. So I think, and I think that's why, yeah, if you're going to keep going, you need to enjoy it. It's the same with, you know, doing exercise or something. If you're just forcing yourself to do it, you can't keep at it because it just takes too much of your willpower. So do something, definitely choose an area, but at the same time be a little bit flexible. So not lock yourself into one specific question. And even if you're really passionate about that question, you might be able to approach it from different angles to better fit in with any kind of funding schemes or something. But yeah, you need to enjoy it. Good.
Great. Well, thank you so much, Gabby, and thanks everyone on the call for joining. Yeah, it's been great. Thank you so much.
Thanks.